Good morning, and I'd like to thank, to start, the organizing committee for inviting me to talk today. I'm very honored to give a talk here at this presentation, and I'm excited to share with you some of our recent research on calorie restriction biology. As you can see in the title, what we are interested in doing are finding gene expression biomarkers of the calorie-restricted state, and then using these biomarkers as a means to identify compounds which mimic the effect of calorie restriction. So as has been shown in several of the previous speakers, calorie restriction is well known to increase lifespan. Here I have some data for mice. That's a fairly long-lived strain of mice that under ad lib food conditions, the mice live to about 40 months of age. So that's a fairly long lifespan for an ad lib mouse. But as Dr. Halsey pointed out last night, a slight increase in the amount of calories provided to the mice leads to about the same amount percentage-wise of increased lifespan. So a 15% of ad lib calorie restriction leads to about a 15% extension of lifespan. And if you can restrict, well, in mice it's very easy to restrict their calorie intake. You only give them fewer calories. So if you can restrict their calorie intake by about 40%, the mice live about 35% longer. So this is a well-known phenomenon in mice and other species that calorie restriction increases maximum lifespan. Not only does it increase lifespan in mice, but also slows the aging process. So many age-related diseases show a slower progression under calorie restriction, they're less later onset. As you can see in this slide, mice that are on a calorie-restricted diet, they visually look younger. So these are mice of the same age, 2 calorie-restricted mice versus 2 mice on a control diet at 40 months of age. So visually, they look younger as well. Now, this is great if you're a mouse, but we're humans, and so what we're interested in is, does the calorie restriction phenomenon apply to humans? Fortunately, there are some studies going on in different locations. One I'm going to talk about is at the University of Wisconsin. It was begun in 1989, looking at a non-human primate. And so it's a study of monkeys given control or an ad lib food intake compared to a calorie-restricted diet. So this is a male monkey who started on the study in 1989. And at the time of this picture, he was 29 years old, near the maximum lifespan for this species in captivity. He doesn't look too bad, perhaps a little overweight. But what's interesting is if you compare that control monkey to a calorie-restricted monkey of the same age, they look just like the mice, I would say. They look visibly younger. And now there's very good data from this study showing that calorie restriction prevents brain-related aging parameters. It prevents age-related muscle loss in this non-human primate. And mice subjected to calorie restriction have a very potent protection against the development of type 2 diabetes. So it seems to be a very powerful phenomenon in a non-human primate that calorie restriction delays the aging process. And actually, it looks right now that there's probably going to be an effect on lifespan in this study as well. All of the control monkeys that were started on this study in 1989, the original group, the most recent death, the last death in the control group was several months ago, and there are 4 CR monkeys still alive in the study. So there looks to be an effect on lifespan as well. Now, the problem with calorie restriction is it looks like this. This is a little extreme, obviously. But, to adhere to calorie restriction, it's not very feasible for humans, even though now it looks like calorie restriction will work. Here's some real-world data from my dinner last night. I would much rather have a diet, and I assume most humans as well would rather have a diet beyond just the lettuce. So a little terrine and cornichon and French bread would be a much better meal. So calorie restriction in humans is probably not going to be an option which can be adhered to robustly. Fortunately, there's a growing field of interest right now looking at calorie restriction mimetics. So identification of compounds, be it drugs or phytochemicals, other nutrients, plant extracts, which have the ability to mimic the physiology of calorie restriction, but without requiring a reduction of calorie intake per se. So this is what our talk is geared towards today, is identifying these compounds which mimic calorie restriction. One approach to use to identify compounds which would mimic calorie restriction is to use transcriptomics or gene expression microarrays, as shown here. As talked about previously, aging and calorie restriction are very complex phenomena, so the advantage of microarrays is that you can study the expression of over 20,000 genes in one experiment. So you can get a pretty clear picture of what is going on with calorie restriction by using a microarray. I would say perhaps it's an unbiased approach because you go into the experiment without a preconceived idea of what's important for calorie restriction. I'm just going to share with you quickly a little data we had published in 2008 where we used microarrays to identify CR mimetic resveratrol. This is a compound found in grape skins and in red wine. And we were interested in seeing if this compound has the ability to mimic calorie restriction. So we put mice on a diet containing either resveratrol at a low dose from 14 to 30 months of age, or we had a group of mice on a calorie-restricted diet from 14 to 30 months of age. Importantly, we did not see an effect of giving mice resveratrol on their body weight, so their body weight was not changed from control mice. Obviously, the CR mice had a lower body weight. And when we looked at the gene expression profile, what I'm showing here is some data from heart. Each dot represents a single gene. And from the 20,000 genes that were identified in the microarray, we found 747 genes changed by both resveratrol and calorie restriction. 745 out of 747 genes were changed in the same direction. So the message here is that mice that were on a 30% calorie-restricted diet for more than half of their life, we achieved a very similar profile by just giving mice the normal amount of food, but a low dose of resveratrol in their food. So we think this is a pretty powerful approach to look at CR mimetics. Now, the concern with using microarrays is that first, they're fairly expensive. Another technical problem with microarrays is that the assay is a little more involved than you would want. If you want to, so let's say we want to look at 1,000, 100, or even 10 compounds for their ability to mimic CR. Using microarrays is just not going to be feasible from a cost perspective or a time perspective. So if we're interested in higher throughput screening, we want to use different technique. Although I said it's great that microarrays can measure thousands of genes in an experiment, and we found 747 changed by CR and resveratrol, perhaps a lot of those genes are specific to the genetic strain of the mouse that was being studied at that time. So we want to find robust biomarkers of CR. We want to get a higher throughput screen using a different approach. So instead of looking at 747 genes that look to be important in calorie restriction and a mimetic, we want to narrow this down to a very small number of genes, much more manageable. So we want to find the best genes that are indicative of calorie restriction. We want to find good, robust biomarkers across different genetic backgrounds, and then we can use these genes as a screening tool. How do we identify which genes are important with calorie restriction? One approach would be to go to the literature. You could do a literature search. And obviously, you would find some genes that many people are publishing on as being important for calorie restriction. One concern I have with this approach, perhaps, is that if there are a few high-profile publications for a given gene being important calorie restriction, what's going to happen? More people will study this gene in response to calorie restriction. And so what we have is sort of maybe a self-perpetuating situation where one gene becomes very important in the field, but maybe a lot of other genes are overlooked. So the approach I'm going to describe today is where we've taken mice from different genetic backgrounds. We've put each of these strains of mice on a calorie-restricted diet, and we let the mice tell us what's important for calorie restriction. So we don't go into the experiment assuming a certain gene is relevant to CR. We do the experiment and let the mice tell us what is important to CR. And I'm calling these calorie restriction supermarkers because they are markers that are relatively consistent across different genetic backgrounds of mice. So we've looked at 7 strains of mice in this study, 6 inbred strains, and 1 F1 hybrid strain. We put groups of mice from each strain on a control or a calorie-restricted diet. We put them on these diets from 8 to 22 weeks of age, approximately 2 to 5 months of age. It's fairly young, but we do see an effect of CR in this time frame. An additional advantage is that we don't have to wait, let's say, 3 years to look at an output like lifespan or long-term calorie restriction effects. So we use a 2 to 5 months age time frame for this study. It's when the mice are adults. And at 22 weeks of age, we collect tissues from the mice. We collect a large bank of tissues. We have them frozen and available for analysis. And what we are doing is looking at microarray analysis between the control and CR groups for each strain. And we want to identify genes that are consistently changed in expression across strains. And what I'm going to be talking about today are the data we have for white adipose tissue. And why we looked at white adipose tissue was briefly talked about last night by some of the speakers. Historically, people have proposed adipose tissue to be an inert storage organ, but within the last 10 or 15 years, there's been a large body of evidence showing that adipose tissue is a secretory or perhaps an endocrine organ. So as was talked about last night, there are factors secreted by the adipose tissue, which in the case of obesity cause an inflammatory state. So some of these factors circulate in the blood, they affect physiology of peripheral tissues, and they have the ability, or they have been linked to age-associated diseases. So there's a link between obesity and age-associated diseases, perhaps through adipose-secreted factors. In calorie restriction, we see the opposite happen, not surprisingly. We see a correction of this adipose tissue secretion, and we see a prevention of these age-associated diseases. So the point I'm trying to make is that we are using adipose tissue because it seems to be an endocrine organ, which has the ability to be modulated under CR and affect peripheral physiology and disease. So here are the data for the 129 strain of mice. As you can see, there are a large segment of genes that were increased in expression in response to CR, and there's a group of genes decreased in expression to CR. One gene was not changed in expression in this strain. This is actually a subset of all of the data, obviously. Again, here are the microarray data lined up for the same genes for the black 6 strain of mice. So what I'm showing you is here are the data for a strain of mice, and each row indicates a particular gene. Again, the data for a bulb C strain of mice, some consistent patterns, perhaps not as many genes changed in expression in this strain. And just to show you the data for all 7 strains, again, this is a subset of all of the data. So from this pool of microarray analysis, we have a large pool of genes that we can look at for ability to be a CR supermarker. What we found in this screen is that there were 414 genes significantly changed in expression in response to CR in 6 out of 7 strains. We saw, I want to say it was 4 genes changed in all 7 strains, but unfortunately the magnitude of expression, or change in expression for those genes was fairly modest, so they would not work well under a PCR-based screening assay. So we investigated genes changed consistently in 6 out of 7 strains. And then we chose 20 of those genes and looked to validate them by PCR in the black 6 strain of mice. This is the strain in which we've done a number of feeding experiments in the past, so we opted to validate these genes in this mouse. And what we came up with was a panel of 11 genes, which we are calling our supermarkers of calorie restriction in adipose tissue. And we want to use these 11 genes as a tool for screening for CR mimetics. Here I've shown you the actual 11 genes that we identified in adipose tissue. What you can see in this figure, each column represents a gene, one of our supermarkers, and each row represents an independent test of different groups of mice on calorie restriction. And what you can see here is that the results from experiment to experiment are pretty consistent. The changes in expression, the fold change in expression, is fairly robust for these markers. In only one case did we not see a gene of CR finding duplicated. This is NAMPT. It was increased as you would predict, but not significantly. However, we opted to keep all of these 11 genes because they look to be good, robust markers. Just to highlight a few of them, CD68 is a macrophage-specific marker, And as we've mentioned previously, obesity is associated with increased inflammation in adipose tissue. Here we're seeing under CR a good marker of decreased inflammation. Not surprisingly, but it's a great marker, I think, to identify a role of inflammation. SERPINE1 is also known as plasminogen activator inhibitor 1. So a decrease in the expression of this gene would be associated with potentially downstream a decrease in platelet aggregation. So here we're seeing a link between adipose tissue and cardiovascular biology. This gene is GLUT4. What this suggests is that in adipose tissue there's an increased uptake of glucose, which might be related to perhaps protection against diabetes. A lot of other markers have to do with adipose tissue biology or adipocyte differentiation. So we have some strong markers, we think, for looking at CR mimetics in adipose tissue. But, are these markers relevant in vivo? Will these markers be useful for identifying CR mimetics? So to get at that question, we performed an experiment where we treated mice in the diet, again from the same 8 to 22 weeks of age, with compounds that have been reported in the literature to have CR mimetic ability. These include bezafibrate, 2-deoxyglucose, 2,4-dinitrophenol, metformin, pioglitazone, resveratrol, and quercetin. So these are compounds which others have reported to be CR mimetics, and we want to do is to see if do our CR supermarkers identify any of these compounds as mimetics, and does that validate our approach? So we fed mice these diets containing these compounds individually. We collected tissues and performed an RT-PCR screen using our markers. I'm calling this our proof of concept study because we want to know if the adipose tissue supermarkers are useful for identifying CR mimetics. I'll show you first the data for bezafibrate. So in each row of this table will be one compound. Each column indicates the ability of that compound to mimic the effect of calorie restriction. So if we look out here, what this means is that bezafibrate had the ability to mimic roughly 100% of the effect of CR for the expression of the SLC2A4 gene. So if GLUT4 expression went from 1 to 2 in calorie-restricted mice, with bezafibrate administration, the expression went from 1 to about 2.1. So 100% means the compound mimicked all of the CR effect. And so the data are shown for each gene. And here, bezafibrate is looking like a great CR mimetic. Actually, I'll get to some complications to this in just a minute. 2-deoxyglucose, in contrast, does not look like a great CR mimetic according to our screen. So only 2 genes were significantly changed in expression by 2-deoxyglucose intake, and only a modest amount of the CR effect was mimicked by the compound. 2,4-dinitrophenol also looks like sort of an intermediate-based CR mimetic. And here are the data for all of the treatments that we gave to the mice. What we did is we tested resveratrol and quercetin at 2 low doses, also at a higher dose. We also tested resveratrol plus quercetin in a combination at the low dose. Just a few things I want to highlight here. Pioglitazone looks to be a fairly good CR mimetic in adipose tissue, and that's probably not surprising because this is an anti-diabetes drug, and under calorie restriction, there's a reduction in adipose tissue size, and this perhaps explains why calorie restriction has an effect on diabetes in mice. So pioglitazone was identified by our screen as probably a good CR mimetic, and I think that's a good thing because it seems to be relevant in vivo. Resveratrol and quercetin at a low dose did not have a great effect on mimicking CR, but the combination actually looked potentially to have synergistic effects. We don't know if that's the case exactly, but when you combine them, they seem to do better than they do individually. So how do we rank these results in terms of which compound is the best CR mimetic? We're looking at a couple of ideas right now. One is considering the number of significant changes that were induced by the treatment. So in this method, we would rank pioglitazone as probably the best CR mimetic. Again, bezafibrate looks great, but I have an idea on that later. Another idea is to look at the average percent of CR mimicry across the genes. So if we take the mathematical average, again, Bezafibrate looks great because overall it's preventing, or it's mimicking about 83% of the effect of calorie restriction. And this is just sort of blindly labeled as the index. It's the product of these two. The reason for including this is in a case where you have roughly a similar amount of average CR mimicry across all the genes, but maybe you want to give some weight or some increased importance to a treatment which is affecting more targets. If you look at this index here, bezafibrate is looking to be the best CR mimetic. What's an interesting case here is that we used a dose of bezafibrate that was reported in the literature as being a CR mimetic, but when we looked at the animal care records for these mice, the diet, I suppose, was actually so bitter that the mice did not even eat the diet. So what we found is that bezafibrate is a great CR mimetic in this study because the mice did not eat the diet. So I guess the point I want to make here is that when you see a report in the literature where someone is talking about a compound having CR-like effects, make sure that the compound is not affecting calorie intake. Otherwise, it's sort of just a positive control. So in summary, what we found are some robust biomarkers of calorie restriction in adipose tissue. We looked across multiple genetic backgrounds of mice, and what we hope then is that there is some relevance to perhaps beyond mice. If we are seeing a consistent effect across different genetic backgrounds, perhaps some of these markers are relevant to humans as well. We were able to use this tool as a screen, and we identified what we would probably call a CR mimetic, pioglitazone. So what we know is pioglitazone has anti-diabetes effects. CR has anti-diabetes effects. So there's a strong proof of our approach, perhaps, if you would say. Surprisingly, we did not see a robust effect of low doses of quercetin or resveratrol. An interesting effect is that we found when you put them together at a low dose, there did seem to be some synergy, and we think this is perhaps another promising way to look in the future, is finding, screening perhaps 100 compounds for CR mimicry and picking the best ones. So just to conclude, what we are looking to do in the future here is take our CR supermarkers and screen large libraries of compounds. And we want to find different compounds, their ability to affect CR, to mimic CR, and perhaps then create formulations from these. And we're doing this in heart and muscle as well with a partner, Pharmanex, to identify combinations of nutrients which might have the ability to mimic CR. And of course, this is still a fairly early stage project because what we don't know is we can mimic CR at the gene expression level, but is aging retarded by an intervention like this? So what we're going to do then in future studies are take the best promising leads from this short-term screen and then feed them to mice for longer-term studies. And then we will look for the ability of this compound or this blend to slow the aging process. And I just wanted to quickly thank this funding agency, the National Institute of Health, for sponsoring this research. Thank you.
Thank you very much. You didn't even have to use the cards. You didn't have to use the time cards. I appreciate that. Very nice presentation. So we have time for discussions. I will pass to a few people in the audience.
Sure.
Just a general overall clarifying concept. In nature, are these non-human primates caloric restricted? Because if you know, I would like to clarify this.
Sure.
Concept. Because you said it's about 29 years or so in nature. Is that correct?
I'm sorry, yeah. So actually, that's the captivity. In captivity, the maximum lifespan is around 40 years for the non-human primate.
So
I don't know what the natural lifespan is.
So we need to know
Right.
What would be the metabolic and
Sure.
dietary situation in nature.
Sure.
And
Just
how that recapitulate your data, I guess.
yes. Like Dr. Halsey showed last night, you know,
Yes.
in nature,
Exactly. The hunter-gatherer.
they are not obese. So,
Yeah.
right.
So I would assume that in nature, in a way, they would recapitulate more your calorie-restricted primates.
Correct.
than
Most likely, yes. Exactly.
okay. So now we have one question there. Okay. So she starts and then you go.
Thank you. So what is the function of the proteins from these genes? Are they all involved in the same pathway? Or do you cluster these genes?
So we do, I have a slide of that, of the gene ontology processes that were modulated across strains. Just one quick comment. That's a very interesting question because we want to know if there's a consistent biological pathway or biological process. But when we intervene by a CR mimetic, we can't screen for the effect of a compound on a pathway. We look at genes. So what we did find is that in terms of pathways, there is a robust decrease in inflammatory gene expression pathways across strains. There was a robust increase in multiple strains for metabolic, mitochondrial metabolism pathways. So I guess what you would expect from what's known in the literature for CR and the effect of biological processes, our genes to some extent replicate or they do agree with the pathway data. So we find a decreased expression of CD68, an inflammatory gene. And we find, one of our supermarkers that I didn't mention was an NAD dehydrogenase, so mitochondrial electron transport gene. Does that answer your question? Okay.
Beautiful piece of work, and I enjoyed it. I think it's urgently needed. I have this question. When you do gene expression profiling, you are looking at genes which are more expressed, but there are genes like some transcription factors whose activity can be also importantly changed, even if the change is 20% or 30%. And you neglect this, but probably there could be many other genes which are very important. And I wonder, for example, about NFKB, for example. And the other question is why you did not consider combination with metformin, which is very interesting from many points of view.
Right.
That's probably better than me.
So to your first point is the transcription factors. I definitely agree with you that there are likely some very important genes changed in expression with CR. Unfortunately, when we want to do a screen of compounds, we look at an RT-PCR-based assay, which requires that the expression of the gene be changed by about twofold to make it reliable from experiment to experiment. So certainly there are some upstream transcriptional cofactors regulating the expression of these genes. And from a biological standpoint, I think it'd be very interesting to mine the data further for those effects. The metformin, interesting. We didn't see it as a great CR mimetic in adipose tissue. Perhaps metformin would be a better CR mimetic if we were to look at liver tissue, where it might have a greater activity. Admittedly, also, we sort of did a simple proof of concept test where we looked at a few compounds. We could expand that as well, and we're also doing that with a large number of other compounds.
Meanwhile, if you here,
Yes.
If your genes change from one strain to another, they're more likely to change from mice to humans. Have you been able to, if one could easily fast people who are going to go to obese and they're going to be treated surgically for 2-3 weeks and then see the change? Have you approached that? And the same thing, fasting. Have you looked or any news from your lab with Rick regarding protein prostration or even methionine prostration, which is claimed to be safe? Thank you.
So we haven't done that exactly, but we do. One of the things I've been doing as sort of a side project is curating what is published for various studies in CR, and we're looking to see commonalities across studies. So we haven't done that with these supermarkers, but that's sort of been on my mind is let's look at different studies and see if our markers show up and see if there's some commonalities between the mice and humans.
To identify supermarkers, don't we need to know a little bit more about fold change, expression level, epigenetic aspects, and most importantly, linking it to the lifespan of the species. So maybe a species with There are mice that have very short lifespans
Right.
that have been extensively studied, like Sam.
Sure. Again, those would be great if, potentially something we could look at as existing data sets for those mice that live a shorter lifespan or mice that live a longer lifespan to see if our markers are recapitulated in those systems. I guess, what we're most interested in is what's most physiologically relevant. And I'm not an expert on the mutant models of mice lifespan, but perhaps they don't mimic what we want to look at in humans. Maybe some aspect of it as well, but we've sort of taken the approach of looking at, well, then again, maybe inbred mice aren't a great example for humans as well, but when we look consistently across backgrounds, perhaps that's most relevant.
Extremely short question, extremely short answer.
Yes?
Is you?
So, apart from those mutant mice, some mice strains have been described that are resistant to caloric restriction. Did you look at the expression of your supermarket of those natural strains?
I believe so if I understand your study correctly, there is one of the strains that we tested that has been reported to not be responsive to CR. And I believe, if I remember correctly, it was one of the strains we tested, and at least in the short term, we did see a gene expression effect in this strain. So I guess I would need to maybe look at those data again for I think it's a DBA strain perhaps, has been reported.